Well, as you can tell, our speaker today is a real rock star. He's professor of biochemistry at the University of Washington, Seattle, and an investigator in the Howard Hughes Medical Institute. He did his undergraduate work at Harvard University and then came here to Berkeley to do his PhD with Randy Schekman. He did postdoctoral work after that at UCSF with David Agard. And since he started at the University of Washington, he's gotten so many awards. He's gotten Young Investigator Awards from almost every place: the Packard Foundation, the National Science Foundation, the Beckman Foundation. He's won the Irving Seigel Young Investigator Award from the Protein Society, the Overton Award from the International Society of Computational Biology. He's a recipient of the Feynman Prize from the Foresight Institute, AAAS Newcomb Cleveland Prize winner, and recently he was elected member of the National Academy of Sciences. So without further ado, David Baker.
All right, thanks, Jay. Well, it's always really fun to come back to Berkeley and see old friends and meet new people, and I've done some of both today, and it's been really great. And so I'm going to tell you a bit about stuff that we've been studying lately in my group. So the key, what we're focused on is the prediction and design of macromolecular structures and interactions. And there are really two problems. The first problem is the prediction problem, which is to go from genome sequence information to the beautiful macromolecular structures and interactions we see in biology. And so this part you might think of as the problem of computing structural biology. You know, there's this tremendous diversity of beautiful structures, beautiful proteins that exist in nature, and in principle, it should be possible to compute their structures from their sequences. In just the same way, RNA structures, one should be able to compute from their sequences. And this is what about half of my group is focused on. The other half is focused on the inverse problem. So while we have tens of thousands or hundreds of thousands of beautiful proteins in nature, there's an even larger set of proteins, macromolecules, that nature never explored. And so this is the design problem. There are all sorts of new structures, new interactions, new enzymes, new endonucleases, new vaccines that potentially exist, that for which amino acid sequences would, for such proteins would be really useful for society, but which we don't know about because nature never explored them. And so the design problem is to go from structure or function back to sequence. And these two problems turn out to be very closely related because in both cases, actually I have this clear on the next slide. So here's a picture. Underlying these efforts, we have a model of the energetics of interactions in biomolecules, and we're constantly striving to make this model better. It's far from perfect. Given such a model, the prediction problem is to go from, say, the sequence of a protein to its structure. We've got the sequence, it's fixed. We're trying to find the lowest energy structure. And the inverse problem, the design problem, is given a structure or function, find the lowest energy sequence. So what's in common to both of these is we're trying to find low energy states. In this case, the lowest energy state of the protein chain given the sequence, and in this case, the lowest energy sequence given the structure. So what this means is we can use the same model for both types of problems. In fact, it's largely the same computer code. And the nice thing is that I should emphasize that our approach is very far from perfect. And the thing that I think is very important with computational models is that you have really critical tests in which they can fail. And because it's such failures that give you the impetus or the insight that you need to improve the model. So the good thing and the bad thing about prediction and design is that they can and do fail. So they're both hard problems. So if you try and predict the structure of a protein, you're almost certainly not going to get it right. At least, and so you can learn from what's not right on how to improve your model, something about the model of the energetics or the optimization, your search for the lowest energy structure wasn't good. Likewise with design, if you try and design a new protein and you go and make it, a lot of the time it won't come out the way you want it to, and so you can learn from that kind of failure. So this gives us a lot of feedback from prediction and design calculations to improve the underlying model. So I'm going to talk about 3 topics today. First, I'm going to tell you about our the current state of our work on protein structure prediction. And the exciting result there is, while we still cannot predict protein structure accurately and consistently over a broad range of proteins, we've reached one milestone, and that's what I'll tell you about. The second thing I'll tell you about on the design side is I'll tell you about designing new enzymes from scratch. And then finally, I'll tell you about a game that we're really excited about that we think could really transform both research and science education. So on the predictions side, the problem one has here is that the space of possibilities is huge. So we start our modeling in all cases with a low resolution model. We're treating the interactions very approximately, but since they're treated approximately, we can search the space very, very rapidly. So this shows a small protein folding up, and the calculation doesn't take much longer than this movie. The main effects are just burial hydrophobics, pairing of beta strands, and so forth. So we can generate such models very rapidly, but and we can generate many, many, many of them, but they are not very accurate, and we can't pick out the ones that are closer to the correct structure from those that are very far away, because we've treated the interactions very approximately at this stage. So in the next stage, we add on side chains onto the model, and at this stage we can compute energies much more accurately because we have all the atoms present. However, it's much more time-consuming it's much harder to move around. There's simply more stuff, and you get collisions between atoms and so forth. So even in this short movie, you got the feeling that it was hard to move around, and that the space explored is much smaller. So what we observed several years ago was that if we carried out many, many independent trajectories, first that low-resolution search, and then the high-resolution search, so we run one movie after another, starting with a different random push, so each trajectory goes out in a different place. This is the result we observed. Each black point corresponds to the end point, or the lowest energy structure sampled during that high-resolution search. And what you see is different trajectories traverse very different low energy structures. So the energies are on this axis, and they're ranging over a broad range, very broad range. Likewise, the structures vary considerably. So this is a case where we know the answer. The true structure would be at 0. And you see that, again, these low energy points not only have a very broad range of energies, but they also have a broad range of RMSDs to the native structure. So if we start with the native structure, that's these blue points here, what we observe typically is that such trajectories not only stay close to the native structure, but they also find much lower energy points or structures than these trajectories starting from an extended chain. So the dominant problem is not so much being able to recognize the native structure when you have it based on its very low energy, but of finding getting close enough to the native structure so the energy drops. I'm showing you an example here where there were a couple trajectories which got lucky and got close enough to the native structure to drop into this minimum, at least partially. And in such cases, if you superimpose these structures on the native structure, here we go. You find that there's quite good agreement. Let's see, I think the native structure is in blue and the predicted structure is in red. There's quite good agreement between the low energy model and the native structure. And what you can see in particular is that side chain packing in the core is reasonably well reproduced. And that's what's producing this drop in energy. It's this sort of jigsaw puzzle-like packing of the side chains in the core of the protein. And the problem is you can only get that jigsaw puzzle-like packing if you're close enough, because if you're not close enough, the side chains can't pack properly and the energy doesn't drop. All right, so I'm not sure if you can see these points, but as I've disposed it, the problem is really one of searching. And the analogy I like is you've landed on some new planet, you have to find the lowest elevation valley, the almost lowest elevation point on the planet. And what we're doing is sending out explorers randomly to explore the planet, and they've got walkie-talkies, and they tell you what the lowest elevation point they found. And obviously, this type of approach, the success of it is really going to depend on the number of explorers. So when we get to a bigger protein like this, which is like a bigger landscape, you see that this initial set of points, we didn't sample, there's nothing really standing out. The lowest energy point is probably out here, very distant from the correct structure. But what we decided to do was recruit computer power from around the world, and we now have a network of we have about 200,000 participants, and what they sign up for the project, we send them the sequence and a starting direction to go searching in, and they search and do the low resolution movie and the high resolution movie, which plays on their screensaver if they have it on. And the graphics are actually much nicer than in the movies I showed here. And then they send us back the lowest energy structure. So now we have very many more dots, even though this is a larger protein. And each dot, again, is the lowest energy point that someone found. So this one could have been found in Moscow, this one in Indonesia. And we actually keep track. So we give special recognition to the people who find, like, this person got lucky, you see. They won the lottery. They have a point that's very much lower energy than anybody else found. And you can see it's actually also not too far away from the native structure. Now, this is also a lead-in to what I'm going to talk about at the end of my talk, which is people watching the screensaver started to write in and say, well, if we could just go in and move it itself and not have the stupid computer move the thing in the wrong direction, we could do a lot better. And so the game we've developed lets people basically interfere with the course of the calculation and go in and play God and say, no, this should go here, this should go there, and then continue it. And so the very interesting question is now with people involved, this is computers alone, what will this look like when people are doing it too? And then will there be some kid in Indonesia who's actually able to get, down here? So so that'll come back to that question. All right, so so there's a question with structure prediction. We've been working on this for a number of years. When do you know you've actually, how do you measure progress? Because it's kind of an incremental thing. You make the energy function better, the sampling better. And one thing there's this test of structure prediction methods called CASP, and one thing that we found in the most recent CASP was that if we started with comparative models generated by draping a sequence over a protein, a homologous protein which had a known structure, and those starting models would be in red, it's sort of dumb comparative modeling. And then we used basically that second movie, that high resolution movie, just to sample around, and did this many, many, many times using all these people's computers. We would reach we end up with a low energy structure which is green, and the blue is the actual structure. So you can see in the cases which I'm showing, which are all relatively small proteins, that we're able to significantly improve the starting templates. And these improved structures were very useful in a way that I will come back to in a moment. Okay, so then the movie I showed was doing de novo structure prediction, so we're starting with an extended chain, and this is an example where there were no homologs of known structure, so we just started with an extended chain, and here was the lowest energy structure found by all these people's computers, and here's the correct structure, and this looks sort of like the picture I showed in one of my first slides of the superposition where, again, the side chains are in roughly the same place. So we weren't too surprised. We were happy to see this kind of thing, but we weren't too surprised. And there were several other cases in Top7 where we saw this degree of accuracy. But the exciting result was that if you took the model, this low energy model, and used it to try to solve the phase problem, you took the diffraction data for this protein. It was solved by X-ray crystallography. And then this model worked very well to phase that diffraction data, so it was just a matter of hours. Actually, Randy Reed was the assessor of these models at CAS7, and he saw our model and said, well, I have written this software called Phaser. I'll get the diffraction data from the PDB, and in a matter of an hour, he had a solution and used automated chain tracing methods to build the structure. So This shows an electron density map computed from the model using the experimental diffraction data and the actual crystal structures inside. So in this case, at least, it was possible to, retrospectively anyway, solve the structure using a computed model, and the refined comparative models I showed you before had the same property that after refinement, one got much better molecular replacement solutions. So there's considerable irony here, of course, because we set out to predict structure and solve the protein folding problem. And what I'm telling you is we're now good enough so that we can help real structural biologists solve their structures faster, which I would like to be able to tell you more than that, but unfortunately I can't at this stage. So in a parallel vein, thinking about NMR, I should say that we had a similar result, which I'm not showing you. If you took NMR models or early stage NMR models and refined them, one got more accurate models, and they were also more powerful in solving the phase problem. Here's a slightly different way to use real structural biologist data. If we take chemical shift data and use them only to bias that very first step in the process, the sort of the building blocks for that low resolution search, but otherwise everything else is the same, the low resolution search itself and then the high resolution search, what we find is that the lowest energy structures we find tend to be very, very similar to structures solved using traditional NMR methods, using NOEs and so forth. And so this is completely blind. So we got from the Structural Genomics Center's data for, this is Yang, Shen, and Ad Bax, who we were collaborating with, got data for these proteins, and we generated models, sent them back to them, and they compared them to the actual structures. And so for small proteins, this method seems to work really well in that with only chemical shifts, one can, and using a fair amount of computing time to find the lowest energy structure of sequences sort of constrained locally by this chemical shift information, one can produce quite good models. Okay, so where are we now? What I've told you is that our models are getting good enough that they can be used in some cases to phase diffraction data. One thing that we're working hard on now is to try and use other sorts of data like lower resolution X-ray or probably what we're really excited about is using low resolution cryo-EM maps to sort of constrain model building. I talked about using limited NMR data, and we're now trying to incorporate for larger proteins where NMR is typically more difficult to incorporate other sorts of data. So basically, the bottom line is we set out to try and predict structure from sequence. We still can't do that, but we think the methods at this stage are good enough to help build reliable models using experimental data, which itself is not sufficient to solve the structure. So it's sort of there's a lot of synergy there. Hopefully, I'll be able to come back here someday and tell you why we don't need the experimental data, but that's not happening anytime soon. All right, so we've extended this now to model membrane proteins and protein DNA, protein small molecule complexes, amyloids, and RNA. And all of the software we develop is freely available, and we're always looking for collaboration. So if you have limited experimental data on some system, and we'd love to try and help build models of that. Okay, so now that's where protein structure prediction, computing structural biology is, and the answer is we're getting better, but we need help still. On the protein design side, this is now the inverse problem, where we're trying to build structure based on sequence, or we're trying to find the lowest energy sequence for a structure. And this shows a movie which explains how this works. Here we have a structure fixed, and we're going to try and find the lowest energy sequence which can adopt this structure. And again, it's using the exact same energy model as I described before, except now we're keeping the structure fixed and varying the amino acids. And this process will eventually find a sequence which, according to the model anyway, is the lowest energy for that structure. A number of years ago, we used this method to we started off with a back of the envelope cartoon of a protein fold which didn't exist in nature. We used this approach to, by iterating between sort of the sequence design and then structure optimization, we were able to come up with a sequence that fit really, really well on that structure. And when the structure was solved, it was found to match very closely to the design model. And so what this showed us is that we could design protein structures with quite high accuracy. We could design brand new protein structures. But the problem, well, by design, this protein doesn't do anything. It really was, it's kind of a boring protein. So what our focus has been on since then is try and design proteins with functions. And there are really 4 different areas that we're focusing on now. The first is the problem of designing new protein-protein interactions, taking 2 proteins that don't interact and designing an interaction surface between them, or taking a target protein and designing a protein that will interact with it. The second area, which I'll get back to in a moment, is the problem of designing enzymes which catalyze novel chemical reactions. The third area is to design very, very specific DNA cutting reagents. And so in particular, we're working on homing endonucleases, which have very extended recognition sequences, and we're trying to redesign these to recognize sites that are mutated in disease. And the idea is that if you can specifically recognize these regions or DNA sequences within about 0.5 kb of these regions which cause disease, if you can simultaneously introduce that enzyme and a wild-type copy of the gene, you can rescue, you might be able to repair the mutation. And then the final area we have a big effort on is designing an HIV vaccine. And here the idea is that the virus has developed this very devious and effective strategy of having these highly variable loops. So when you get infected by the virus, you mount an immune response to the loops, but not to the few key regions of the virus. So the virus does have some critical regions, like I like to think of them as Achilles' heel, where it's weak. For example, the CD4 binding site needs to get into cells. But the problem is they're kind of hidden away, so your immune system doesn't make a very strong response to them. So what we're trying to do is design small proteins which present these critical regions up front and center to the immune system. At this point we have designs that mimic parts of the virus. When you put them into animals, our collaborators do this, they produce antibodies which recognize the epitopes. So far they aren't neutralizing. If they were neutralizing, that's probably what I would be talking about instead. So, along with solving the protein folding problem in a couple of years from now. So, all right, so our goal, I'm going to get back, focus now on enzyme design. So the goal is to create, the long-range goal is to create enzymes which catalyze reactions not catalyzed by naturally occurring enzymes. So there are obviously a wide range of important and useful applications for enzymes in cases where, for applications where enzymes currently don't work. So difficult problems in synthetic chemistry, something close to all of your hearts is making new fuels and so forth. This is also a critical test of our understanding of how naturally occurring enzymes work and a grand challenge for computational protein design. Okay, so what's the approach? So basically, we're going to proceed in the opposite way as you learn about enzymes in a biochemistry class. You get the structure, you see the structure of the protein, then you see the blow of the active site, and you see all the detailed interactions with the transition state or substrate. We're going to do the opposite. We're going to start by computing the transition state, and then we're going to design an ideal, or in principle, ideal active site around this transition state. We're going to position functional groups, just disembodied ends of side chains, say a negative charge here, a positive charge here, an OH group here, around that transition state so as to mediate catalysis. After we've designed that ideal active site, the next step is to design a protein which has this ideal active site. I'm going to take you through how this works. Okay, so the first reaction I'm going to talk about is the Kemp elimination reaction. And here, it's a very simple reaction where we're going to break this nitrogen-oxygen bond. And the way that we're going to do it is by abstracting this proton here from the substrate. And here in this simple mechanism is the transition state. So what we need here, so what do we need to do? We first need to decide what the constellation of amino acids, functional groups that we're going to try to use to catalyze this reaction. So that's the first part. Choose the catalytic motif. And we chose two. These are so for the choice of the base, on the left, we've got a carboxylate group right here, abstracting the proton. And to stabilize this negative charge here, we have a lysine. In the other choice that we made in this case is a histidine backed up by an aspartate lined up to abstract this proton. And in both cases, we have an aromatic ring packed on the substrate, which will facilitate binding and electron delocalization. So this is sort of, this picture really, the only thing we're specifying is that we want a carboxylate group here and an amino group here, for example, in this case, and a ring here. We don't specify where any of these atoms have to be. So now what we need to do is build a protein which has such a site. And what we did in this case was take a library of about 100 thermophilic protein scaffolds, take away all their side chains and scan through them to see if there's a place you could fit this, this active site so that these residues, so that the transition state would fit in and these residues would make the interactions they're making in the picture. All right, so that process is shown here. We have a variety of different scaffolds. We're going to find places where we can actually reconstitute this site. And so so we have an algorithm which I probably don't have time to go into, which can rapidly scan through a large number of structures, identifying backbone positions where you can place these types of residues and such that all these interactions can be realized and the transition state fits. So the next step, so here's these three residues here. Here's the glutamate, here's a pi stacking residue, here's the transition state. The next step is to use the algorithm that I showed in that design movie. We let all the side chains in the neighborhood vary to maximize binding to the transition state. And that, at the end of the day, we have designs that look something like this. Here we have, I don't know, can you see this all right? Is there a way of turning down the lights just a bit? Okay, so this is, that's probably too much, but we'll come back to that. So anyway, so here we have the first motif with the glutamate abstracting the proton from the transition state, which is underneath. You see there's a hydrophobic pocket, which the transition state model fits into. You have the aromatic residue on top, and on the right, you have this asp-his mechanism where the his is abstracting the proton again from the transition state, and you have the aromatic pocket and then the ring on top. We better have the lights on again, otherwise people will fall asleep. Okay, so now the exciting thing, so okay, what do we do then? Well, it's kind of neat. One very easy and not easy to do at all when I was a graduate student here, but now it's easy to do, take these amino acid sequences. You can obviously turn them into DNA sequences, and you can either assemble a synthetic gene in the lab, or you can send the sequence to a company and they can make a synthetic gene. So, so we ordered, in this case, we ordered synthetic genes, and they come back and they're in E. coli expression vectors. So it's basically a one-day purification, and then you do an enzyme assay and you cross your fingers. So in the interest of full disclosure, most of our designs didn't work. I'm going to show you the results on the ones that worked. So the one take-home message you should take from my talk is computational methods are far from perfect, but they can get you in the ballpark. So here is the design on the left, the model on the left that used the glutamate. You see there's activity. This is just product versus time, and when we mutate that that glutamate that was the base to a glutamine, we completely lose activity, which is good. So this thing is catalyzing the reaction for the reason we think it is. And here's the reaction on the right, where we have the histine mechanism. And when we mutate the histine to an alanine, we completely lose activity. When we mutate that aspartate that was positioning the histine to an asparagine, we lose some activity. So these mutational results are consistent with the design working for the reason we think they do. In a couple of cases, we have crystal structures, and the caveat is we don't have crystal structures with transition state analogs actually in there, so we just have to model them in. But you can see the backbone's basically in the right place, and the side chains are basically in the right place. And this has been true for all of the structures that we've been able to solve. All right, so how do we figure out what's missing, and how do we make them better? Well, we'd obviously like to make them better in the long run by improving our design methods. But in the shorter term, one route to that is molecular evolution. We collaborated with Danny Tofik and Olga Karansky, who basically made random, we gave them the design, an early design, and they made random variants and simply screened for those variants with increased activities. And the really neat result was, here's the design. This is one that I haven't shown you yet. Here's the transition state model, the catalytic residues. Here's the inner site in yellow. And the mutations they found are in blue. For the most part, they're sort of in the second or third shell. And I think there's an important point here. The computational design can kind of get the broad outlines of the active site, right? But there are a lot of subtleties having to do with positioning of backbone elements, maybe dynamics and so forth, things that we can't really control so well in the design process. And by this screening approach, one can identify mutations. And in a number of cases, we actually now understand what's going on. In some cases, they're fixing foolish problems with the design, initial design, that we should have realized in advance. In other cases, there are more subtle effects we have to work to incorporate in the next generation of methods. All right, so here are some numbers. The things that you want to look at are probably in these two columns. So this reaction does go on spontaneously with a non-zero rate. The rate enhancement by these designs is on the order of 5 orders of magnitude. This may seem a lot, but naturally occurring enzymes can do this can enhance rates by 17 orders of magnitude. So we're far from what naturally occurring enzymes can do. The best evolved enzyme is now a variant of this one, which I don't have on here. It has a, this is kcat over Kemp. So naturally occurring enzymes typically have kcat over Kemps around 10 to the 6th. It's close to diffusion limited. That's the point they've been optimized to. You can see we're far below that. The best one we have now is about is in the 10 to the 4th range. So we're getting closer to respectability. But really, this is sort of first baby step in this area. Okay, I'm going to tell you about one more example. This is a carbon-carbon bond breaking reaction. This is the bond that's getting broken. This is a retroaldol reaction, and it's going to proceed through a lysine that makes a covalent covalent bond with the substrate. So this is the previous reaction was using general acid-base catalysis, and here we're using covalent catalysis. So this is a complicated slide. The main point is that there are many steps in this reaction, and one really has to not just think about one transition state, but think about all the different steps. And it's important that the enzyme, that your design be at least compatible with all the steps. And so one thing I'll get back to is I think enzymes really have these huge compromises they have to make, because stabilizing one step may be very different from stabilizing another. Let's see. So at different stages, one wants a base, for example, to abstract the proton from the alcohol. One wants a hydrogen bonding group at this stage. So there are different requirements at different stages. So remember, the first step in our design process is to come up with catalytic motifs. And we tried 4 different ones in this case. The first one we tried was inspired by naturally occurring aldolases, which use a complicated salt bridge network. It's not exactly what's shown here, but this is loosely inspired by it. You have a lyse, asp-lyse, and the sort of protein shuffling that's going on along here. The second one is really radically different. It's very simplified. We just have a lysine forming a shift base here, and then a tyrosine interacting with the alcohol. Here we have something a little more sophisticated, an asp-pis dyad, and we use this over and over again in our designs. You saw the first one in the Kemp to abstract the proton, and again the lysine forming the shift base. And the final one's a little bit different, because what we do is we try to position a water explicitly. We try and bind a water molecule so that it can help in this proton shuffling between the proton from here and here. And we do that by way of 2 hydrogen bond two side chains which we placed to interact with the water. Again, we have the lysine forming the Schiff base. And again, we use the same algorithm to search through scaffolds for a place where we can build this site. We find many different scaffolds. I should say one thing that was very interesting for the Kemp is that we over and over again found matches in Tim barrel proteins. And the reason in that case was pretty clear, is that the substrate fit quite nicely in the middle of in the sort of the typical site, binding site of the Tim barrel. And Tim barrels have so many, all the different strands have C alpha, C beta vectors that point to the active site. So there are a lot of different opportunities for these catalytic residues to come off. So I think we may have, to some extent, recapitulated what one sees in nature with the diversification of functions of the Tim barrel proteins. We get matches in all these different scaffolds. We get, and that's basically shown here. And the first result when we started experimentally testing things is the first two mechanisms just didn't work at all. We made a number of designs and we got no activity. It was only when we went to the design that involved the explicit water molecule that we started getting any detectable activity. Okay, oops, let me go back one, sorry. Let's see. So this is, I'm just going to show you examples of design models for a couple of these. And do the lights need to go out, or is it? Yes? Okay, how did you turn the lights off? So here we have the water molecule being positioned between two hydrogen bonding side chains. Here's the lysine forming the Schiff base. Here is a hydrophobic pocket which binds the aromatic part of the ligand. And here's another design. Here's a water molecule again being positioned by two side chains, a lysine coming in, and a hydrophobic pocket for the ligand. And it's actually quite non-trivial to be able to place so many residues simultaneously and actually form a cavity too. So the main algorithmic limitation we have is if you wanted to specify 3 more things, we simply wouldn't be able to find any place in any existing scaffold to do it, and we'd have to build the scaffold from scratch using the types of techniques I talked about earlier. So, again, we have crystal structures of a number of these designs. In this case, this tryptophan has moved a bit, but again, we don't have the transition state bound. Okay, let's turn the light back on. Okay, now again, we have, I'm showing you the cases that worked, which were a minority, as you've seen already. Here's activity versus time. This, you see this enzyme, or protoenzyme, whatever you want to call it, there's initial burst phase, and then it slows down. It turns out this is due to product inhibition. The product can go back in and inhibit the enzyme. If we change the lysine to a methionine, we completely lose activity. If we mutate one of the residues that's supposed to be coordinating the water, we lose activity, some activity as well. The same thing is true in the other design I showed, that here's the activity of the, quote, wild type design. If we mutate the lysine to methionine, we lose activity. If we mutate the threonine to a glutamate that was making a hydrogen bond, we lose a little bit of activity. All right, so what can we conclude from this? So we can design enzymes, although it's a little dangerous to call these enzymes because they're really not very active. The level of activity of these retroaldol designs is similar to that of the Kemp ones. The Kcat over Kms are actually even worse. We can do this repeatedly. As I told you for the retroaldolase, it's kind of interesting that this mechanism that used an explicit water was more successful than the other mechanisms. And you can imagine this is because say that first mechanism where we had 3 buried charged residues, there's a huge desolvation cost to burying charged residues. And to do that successfully, you have to satisfy all their hydrogen bond donor and acceptor capabilities with other networks. And so, but designing these extended networks of salt bridges like you see in naturally occurring enzymes is really challenging. And we sort of are cheating in this case. We're substituting a positioned water molecule for some of that machinery. And it's tempting to speculate that early in enzyme evolution, similar compromises may have been made, that it just was too hard to it was very unlikely to simultaneously get a beautiful catalytic machinery in place all at the same time. So rather, you might have had mechanisms which made more use of water, and then over time they would be replaced by more finely tuned side chain networks. And we're trying to design enzymes for other reactions. And so I want to emphasize that while we can design things that have activity, we're really far still from where naturally occurring enzymes are. And I think it's a question which I am lying awake at night thinking about is what do we have to incorporate, what principles do we have to incorporate to increase to design enzymes with rates that start approaching those of the exquisitely optimized naturally occurring enzymes. And my current guess is the key is to position the substrates as precisely as possible relative to the catalytic machinery by putting handles on them that the enzyme can grab onto to do that positioning. But I don't know. That's what we're currently trying. And again, this idea that enzymes are the masters of the art of compromise, and you really appreciate this when you try and design things, you focus on a single step. And in fact, in some of our early designs, we made things that we then realized couldn't work because they were completely incompatible with another step. So what enzymes do is really marvelous. They have to compromise, be really optimal for a particular step, but still be compatible with all the other steps. All right, so I'm really privileged to have a really talented coworkers, and usually I go through and acknowledge them as I go through, but I somehow didn't start off doing this. So Riju Das, really talented postdoc, who I think some of you heard when he was here, extended the methodology to RNA structure prediction, and he also carried out the de novo structure prediction that resulted in the molecular replacement test. Phil Bradley, who you also heard a couple years ago, really developed a lot of the sort of the overall protocol I described for high-resolution structure prediction. Randy Reed did the molecular replacement. Ad Bax and Yang Shen we collaborated with to do this, to use chemical shifts to try and build structures. Not on this list, but they should be, are Bin Chen, who did the homology model refinement, and Vatsan Raman, who did the NMR structure refinement. On the enzyme design side, Daniela Roethlisberger and Andrew Wallacott did the work on the Kemp elimination. I told you that Danny Tofik and Olga Karansky, his student, did the molecular evolution. Alex Zangalini developed the computational matching methods, and Ken Hauk and Jason Duchansky did some of the initial quantum chemistry calculations. For the retroaldolase, Eric Althoff, a really talented postdoc, and Lin Zhang, a very talented student, worked on that. Barry Stoddard solved the crystal structure that I showed you. And again, Ken and his postdoc, Fernando, did the quantum chemistry. All right, so now in my last 5 minutes, I get to come to the game. This is the work of two really exceptional graduate students, Adrian Troy and Seth Cooper, who are working between me. They're computer science students. They're working between my group and Zoran Popovic, a professor in computer science, and that's what I'm going to get to now. So as the students, as the people who've been paying attention to this know, we posted, well, I'll get to the game. So I have bad news. I'm afraid I have bad news for all of you. It's very disappointing. I'm deeply disappointed. We have this, we have made a challenge. I'll show you the game in just a moment, but it's a multiplayer competitive game, and we set up an initial challenge between UC Berkeley and University of Illinois, and I'm happy to say, even though it's not on here, that Berkeley won the first challenge, but unfortunately Illinois appears to be better in the long run as they've won in both Challenge 2 and Challenge 3. But you know, what can I say? So let's just find out quickly who the top scorer is from, or actually, well, we'll come back to that. I have time. Okay, I should show you the game. All right, so here's the game. So the goal, so our question here, remember, we had these people looking at the screensaver thinking that they could go in and make things better. So we've tried to make that possible. So what you can do in this game is it's basically just an interface to Rosetta, the program that everything I've described so far is done with, except you can go in And you can do things. You can pull things around. You should be able to pull things around, although it doesn't want. There's some sound effects. Here's my score up here. I can look and see how I'm doing compared to other people. I'm now ranked number 26 in the world, which isn't really so bad. Let's see. There I am, you see. OK. Now watch me work my way up the. See, I should have signed up for Berkeley. Then you guys would have lost for sure. OK. So now I'm going to optimize the side chains. That didn't do too much. Now, and that this actually, I should show that a little bit better. OK, let's just move this side chain. So you see that red thing. That red thing was a clash. That's bad. We're trying to. So the key is how do you describe this in a way that people who know nothing about science can appreciate? I'm not doing a very good job here. OK. Now this shake side chains is exactly what that movie was doing. Watch if you see this. See it? Well, it was reconfiguring a lot of the amino acids. Now the wiggling is now I'm just going to. It's a fancy name for minimization. Now it's going to try and it's using Rosetta now to try to optimize the structure. Look, now I'm up at 89. I'm doing pretty well here, and we can see if I've moved up. Does it have my latest score? I know. Oh, that must be my score. Oh, it should update this if I'm on the internet. So I think it should tell me that. Let's see. Oh yeah, stop the wiggle. I have to stop. OK. So now I'm at. I've moved up. Have I moved up? Oh no, I haven't. Oh, that's too bad. Well, OK. It's tough. I'm really not very good at this, but. Anyway, so I hope you had fun with this. What we're so what we're hoping is that the game is getting to the point where it's fun enough that people will actually pick it up. My son, this was also inspired by the fact that my kids in Seattle Public Schools have the world's most boring science classes. So we're thinking, how can we make this more fun? And so at this point, what I'm pretty convinced of is that this game can be useful for you don't normally grapple with macromolecules in your ordinary life. So here you literally grapple with them, I can tell you. For those of you who've played it know that you grapple with them more than you probably want to. But we're also really excited to see whether people working with computers can solve hard problems that computers can't solve alone. So like in that example at the beginning, if we send out that sequence and now people can do it together with their computer, can they find lower energy structures than the computer can alone? And I sort of have this dream that there'll be some 12-year-old in Indonesia who can just see proteins in their head. If you've ever played Halo, it's a lot harder than this. I mean, I tried playing with my son, and it was like incredibly difficult. So they'll just be able to guide the protein in. The other thing we want to start sending out soon are the design problems, the things. And one of the problems would be, for example, you see the influenza virus, the flu virus, and you see a protein docked up against it. And now you can use all the tools I showed here. Plus, you can remodel, grow out loops, add extensions on. Your goal is to get the tightest binding thing to the virus you can. So your score here is you get a higher score if you have a tighter binder. So you come going to work the next morning, people will say what you did, what did you do? You don't say you played a computer game. You said, well, I was curing the common cold. Well, then we get all of these sequences back, and we can look at the lowest energy models, and we actually experimentally test the ones that look most promising. So anyway, so we're quite excited about the idea of sort of going beyond using people's computer power and trying to use their brain power as well. And I think if you get people all around the world working to solve some of these problems together, I think it can be pretty neat too. So anyway, I'll stop there.
So, I mean, this game, it sounds like so much fun. I'm going to go home and start.
Good.
But
I think
I'm having a little bit of trouble believing that I can really have my hands on all the forces from this particular type of picture. In particular, the protein is sitting in some kind of environment. Maybe it's membrane bound, maybe it's a solvent, whatever. And those molecules
Yeah, so I didn't,
It's sort of like I have to play with this in a bathtub or something.
right.
It's a bathtub.
Well, no, you sort of have the bathtub here, in a way. These things that are glowing, these things are hydrophobic, and these are polar. We've got an implicit solvent model, so it's got all the disadvantages and inaccuracies of an implicit solvent model, but at least the solvent is being modeled, at least not discretely. And as I showed you, the energy function's accurately enough. It's basically exactly the same energy function I've been describing all the way along, with all its strengths and weaknesses. So as far as whether, I mean, so there are two issues. One is the model's incomplete because we're not modeling, for example, the discrete nature of the solvent. The other is, how does the person appreciate, you know, what a van der Waals interaction is like from the game? And to some extent, what the person's doing is doing lower resolution stuff. They're saying, well, maybe if this helix goes over here, okay, and the computer's doing a lot of the heavy lifting. The computer's doing all the sidechain optimization and the heavy-duty minimization. So the person's kind of providing more of the heuristics. Like in the movies I showed at the beginning, basically it's a Monte Carlo with minimization process where the computer makes a random move and then there's minimization. Here, you can replace the computer's random move with a person's move, and all you have to do is be a little better than random to perhaps beat the computer.
So the retro-alkylase, you said you got to worry about transition states for all those intermediates.
Yeah.
But you really don't. You only have to consider up to the first irreversible step, and which in that case is the rate determining step. It's carbon-carbon bond movement. So that's really.
But even up to that point, right, you have to form the carbanolamine. There are some steps, yeah.
The rate determining transition states.
But then you have to recycle for the second round, right? You have to get the product off.
But for the molecules you've chosen, that would be fast.
Well, we get product inhibition. But you're right. I mean, we don't have to consider absolutely every step, but we still have to consider more than one step. Yeah.
So you started with a number of thermophilic scaffolds.
Yeah.
So what happens if you try these enzymes at high temperature? What happens if you try mesophilic scaffolds for your?
Yeah, that's a really good question. So they weren't all thermophilic. We wanted proteins where we could go in and make 20 mutations and they'd still be stable and fold. The question about high temperature, after talking with Judith today, that's a good question. We haven't looked at the activity at higher temperature. We've done some designs on, not all of the designs are on thermophilic scaffolds. But the answer is we really haven't explored that at all. You know, I hadn't really thought about that.
So decades ago, people used the combinatorial machinery of the immune system to
Yeah.
design catalytic antibodies.
Yeah.
Can your design tools sort of verify that those catalytic antibodies work for the same sorts of reactions that?
Well, let's see. So catalytic antibodies have been generated for these reactions, and there I think it's almost, they come at, in catalytic antibodies you're selecting for tight binding. And whereas we're trying to optimize positioning of the key residues. So it's kind of like you want to put the two together. Because I don't think we're binding our transition states models tightly enough in the right way. So I don't think there's so much that we can verify what they're doing, but we can certainly learn in the sense of, one of the things we're trying to do in collaboration with Don Hilvert is take these designs and then try and enhance binding using techniques like yeast display. So we can learn from some of the things that people did with catalytic antibodies.
So two questions. One, does the phasing field converge if the model's wrong, and where is that threshold? So could you just throw models at it until it converges? And then the second was, have you gotten Alexei Mersin to participate?
Okay, yeah. So on the second, you know, I was looking for his email 2 nights ago with exactly that question in mind. For those of you who don't know, this thing is now public. You can get it. The URL is very clever. So you can, so I haven't gotten Alexei Mersin, but on your recommendation, I'll go back and look for his email again. We can, in fact, you're right. So when we're doing collaborations with people, we often ask them for the diffraction data because then we can screen through many models. Yeah, and that. But the answer is, as people know who've tried to phase diffraction data with comparative models or with NMR structures, you have to be really close before you get a solution.
The Rosetta code open source, or do you, if it's not, do you foresee it becoming open source?
So the code now is free to academics. Anyone can get it and do anything they want with it, and we like to hear about it. We have this annual developers meeting, so if people get the code and do something with it, then they're invited to come back. And the reason why it's not completely open source is we license it to companies, and what that money goes to is to pay anyone in the world plane ticket and conference fees for this Rosetta meeting. So it's very important that we have an annual meeting where all the developers come to, and that's the way we support it.
I'm not sure you've done this, but if you, presumably for any protein that you design, you can look at it, the effects of mutations on the structure.
Yeah.
If you compare your purely designed proteins, where you take some artificial scaffolds to naturally occurring proteins, do they have a similar, do you see some kind of difference in how you predict mutations affect them?
Well, there is a difference. I mean, that top 7 protein I showed was just radically stable because we had optimized it just to be stable. It had all the perfectly packed hydrophobic core. It didn't have any imperfections in stability, no compromises that you need to have for stability to get function. So it didn't have any catalytic sites. So that was really the remarkable feature. In terms of the effects of mutations, though, I guess the other unusual thing about top 7 is that its folding was quite non-cooperative. Very different from naturally occurring enzyme proteins. But the effects of mutations were what you'd expect. If you removed a hydrophobic group in the core, you destabilized it.
And so there's nothing in looking at those design proteins which we're able to suggest that there's something about how the evolvability of proteins, something about the evolvability of naturally occurring proteins or anything like that.
Well, I think the real key question is how stable a protein is. The more stable a protein is, the more insensitive to mutations, because the more you have to destabilize it the more it can tolerate. And that's what we saw, I think. Yeah.
Do you think the flexibility of enzymes is important for sensitivity? Do you plan to use?
Well, we should ask Judith that question. We talked about that earlier. Yes, the answer is yes. But we aren't really so I had that on one of my slides, the question about dynamics and flexibility. And so we clearly are designing very poor catalysts. Why are they not good? Well, it could be that we don't have the right dynamics in there. That's certainly one possibility.
Okay, we want to thank our speakers.